Hello, and thank you for joining our Morning Commute podcast series on multiple sclerosis. Morning Commute is developed in collaboration with At Point of Care and Projects in Knowledge and is part of a continuing medical education series. This CME CE activity is supported by an educational grant from Biogen, Bristol Myers Squibb, and Sanofi Genzyme. Information about the faculty and disclosures can be found at morningcommutepodcast.com/MS6. You can use this link to receive your credit and evaluate this program. The URL can also be accessed in the episode notes. You can also find the complete 6-part series by visiting morningcommutepodcast.com/MS. This is the last episode in our 6-part series, Eyes on the Horizon. MS disease-modifying therapies are an evolving field with new therapies and new diagnostic tools. Our faculty will take a look at where we are now and what the future holds as new MS therapies emerge. For MS, has the era of true personalized medicine arrived? I'm your host, Candace Hoffman, Managing Editor of Morning Commute. We are joined by Dr. Fred Lublin, who is a Saunders Family Professor of Neurology and Director of the Corinne Goldsmith Dickinson Center for Multiple Sclerosis at Mount Sinai Medical Center in New York City. And by Dr. Robert Bermel, a neurologist in the Neurological Institute's Mellon Center for Multiple Sclerosis at the Cleveland Clinic in Ohio. Dr. Lublin will begin our discussion.
Welcome to our podcast, Eyes on the Horizon. I'm joined by my colleague and friend, Dr. Rob Bermel from the Cleveland Clinic. Welcome, Rob.
Oh, thanks. It's so great to talk to you today, Fred.
So in this podcast, we're going to look a little to the future. And before we can do that, we need to look at the present and say, what are our current needs? We've made some remarkable strides in MS. It's at the leading edge of neurotherapeutics and has been for the last 20-some years, where we now have over 20 disease-modifying therapies. And we're proud of that, but we're not satisfied. So we have a number of needs moving forward as we look to the future. Things like I was starting out with, we have all of these therapies for relapsing forms of MS, but we still could use better agents and a combination of better agents and safer agents at the same time, and having long-term safety information. Rob, where do you see us going? What else are our major needs?
Well, clearly we have a need for therapies that slow down the progression of MS. I would put that down as probably one of the greatest needs for patients who come into our clinic day after day, and that we have great therapies right now for limiting acute inflammation, that is stopping new MRI lesions and stopping relapses. But for patients with progressive forms of MS, there really still exists a need out there, and probably a belief that this is going to involve tapping into different mechanisms than the ones that we're currently utilizing that largely affect the adaptive immune system. The other big need, I would say, is for repair strategies. So I would talk about therapies that would have the potential to augment repair mechanisms in the nervous system, or potentially augment remyelination efforts. And the holy grail of all things neuroscience would probably be to try to restore function. I know it's a major mission of the National MS Society in the U.S., and it's also where a lot of the research dollars and brain power are focused these days.
Well, I think that lays it out nicely. You mentioned one thing I want to explore a little more, because you mentioned looking for agents that affect the innate immune system, which we're going to talk about in a little bit. What about more purely neuroprotective molecules?
Well, for sure. I think I've always used the analogy that, you think about protecting a house in a storm like a hurricane, and there's really a couple of different ways that you could do that. One would be that you put hurricane shutters on the house, so the hurricane's going to come by, but the house is going to be protected. Storm's going to blow past, and then you can take the shutters off and everything will be okay. The other way to do things would be if you had some sort of a weather machine that could prevent the attack to begin with. So the treatments that we have right now really amount to that weather machine. They are neuroprotective in a way, if you think of it, because they prevent acute immune attacks on the nervous system, preventing enhancing lesions, preventing MS relapses and attacks of acute inflammation. But it would be nice if we had that other mechanism also, especially since there's probably smoldering inflammation that goes on, and especially, as you mentioned, Fred, potentially with the innate immune system at play. And so having therapies that act directly in the nervous system to protect axons and protect myelin that is more of a hurricane shutter type of mechanism would be really useful. We need to evolve first measures for how these might work. And we've seen some pretty cool trial designs in recent years where we're beginning to test in early phase trials medications which have the potential to protect the nervous system. So I'm thinking of trials that may use advanced imaging measures as an outcome, or in some circumstances might use something like a model system. For instance, acute optic neuritis or chronic optic neuropathy, one particular functional subsystem of the nervous system, as a model in order to test these things. And we've already had some early successes, right? So one of the molecules that's been tested, a drug called Ibudilast, which was run by a trial led by my colleague Bob Fox here from Cleveland Clinic. It was an international multi-center trial of this drug called Ibudilast, which is a multiple different mechanisms of action, but is classified as a phosphodiesterase inhibitor and used in Japan to treat patients with asthma, but repurposed in this trial using brain atrophy, brain volume as an outcome measure actually in the trial. And in this multi-center phase 2 trial actually did show that it reduced brain atrophy in patients with progressive MS, either secondary progressive MS or primary progressive MS, by about 50% as measured by brain parenchymal fraction, again, in this small early phase trial. And so if we could begin to design trials with advanced imaging methods, like looking at brain atrophy, or in the case of Ibudilast, there were actually other imaging measures that tracked along with brain atrophy, things like magnetization transfer imaging or OCT, optical coherence tomography, we'd begin to develop a pipeline for how to test these neuroprotective drugs. Another success story I would highlight is the medication Clemistine, which was tested by the group at UCSF out of a large medication repurposing effort, rapid throughput screening of molecules which may have the potential to spur remyelination. And there in a visual system model, actually appeared, when modeled electrically using evoked potentials, appeared to have an effect on remyelination when patients were randomized to Clemistine versus placebo. So again, I think the clinical trial methodologies are evolving. I think we have a couple of very notable success stories, and I'm hopeful that this will lead to further developments in the field of neuroprotection.
In MS therapies, there are those that seek to repair and those that offer neuroprotection. Doctors Loveland and Bermel turn to the issue of repair with a look at Ibudilast and the anti-LINGO trials that studied opicinumab. Let's rejoin our discussion.
So actually, I like to separate out repair from neuroprotection, because I think they're different. I actually think they should be studied differently, because I worry about washing out an effect by asking a drug to do more than it ought to be. So when I think about repair, I think about something that does just that either remyelinates, or one would hope in the future could generate regrowth of axons. At the present time, remyelination seems the more likely goal. Although back when I was a boy in medical school, we were told that central myelin didn't repair. We now know that that's not true. We also were told that all the neurons that were in your head the day you were born were all you're going to get. And we know that that's not true either. So things change, and these things happily change. But I like to think of them separately. And I think that molecularly, we've been stymied on the idea of neuroprotection as a true neuroprotective thing, looking at NMDA and other molecules that may be involved in cell death, separate from, and I'm going to come back to this idea of the innate immune system. But I think that in that regard, MS is more like Parkinson's and Alzheimer's and ALS and things like that, where there's tissue dying off. and where we badly need to have some strategies to protect that dying tissue. And it's failed. It's mostly failed because they've been testing it in diseases that are much more difficult to make a dent in, including Alzheimer's and true disease modification in Parkinson's, which hasn't happened yet, or certainly ALS, which is extremely difficult to treat. And so a lot of good molecules have gone down the path. But I think that's a separate approach. And I'm still waiting for some real molecules to get at that. The ibutalas was a little close, although it was still phosphodiesterase. It's kind of more sort of immune, semi-immune, but had some interesting effects. And certainly that was an important and nice study. But in terms of the repair, then I think about the efforts that you were discussing that we've made with remyelination, like clemastine and molecules like that. But we also have the issue of LINGO. So LINGO was a known mechanism for inhibiting a repair, and the anti-LINGO approach, which has been difficult, and their latest study was unsuccessful. But I'm hoping that doesn't have the same effect as some of these unsuccessful neuroprotective trials have had in other diseases, because it's an important area, and that's an important mechanism. And so what they were doing there is, to me, an order of magnitude more complicated than what we're doing with our disease-modifying therapies in the anti-inflammatory group, because we don't know there what the timing was. One interesting aspect of that was that they took a different approach than the clemastine group, looked at individuals who had chronic demyelination. They were waiting a time, whereas in the in the anti-LINGO, they were going for more acute demyelination. And it's an interesting argument. If you wait too long, do you then put the underlying axon at risk and you'll never bring it back, versus trying to separate out the repair that happens naturally after an acute attack. The other problem with anti-LINGO, while the target engagement was an interesting one, doing it with a monoclonal antibody to try and get into the central nervous system is a little difficult also.
Yeah, for sure. I know in the upacinumab trials, the anti-LINGO trials, they wound up having to use a very high dose of that monoclonal antibody, because it is challenging to think, how something dosed systemically in a large monoclonal antibody would potentially have efficacy in the CNS. This idea of repair, though, in remyelination is it's inherently appealing, because we know from pathological studies that oligodendrocyte precursor cells exist in abundance in the brain, and that in fact remyelination does occur, especially in early acute MS lesions. So you could definitely see using electron microscopy or even immunohistochemical stains with light microscopy, the remyelination of axons happening. But like you said, it's such a time-sensitive phenomenon. That axon is at risk if it's exposed, and the faster you can get it remyelinated, the better. Everyone's hopeful that the potential does exist, but the exact mechanisms and how to attack it, I think the science is still evolving. I mean, I think there are, thankfully, animal models now. So animal models of demyelination and remyelination, such as a modified cuprizone model that's become popular in a lot of labs, that hopefully is going to spur the development of some of these newly discovered pathways and new mechanisms. Even when trials like the apacinumab or the anti-LINGO trials don't result in a drug that's going to market, I'm really hopeful that we learn something from them. And so one of the things, for instance, that we learned from the anti-LINGO trials and that acute optic neuritis model that you're talking about is that the nerve cell loss, the ganglion cell loss, occurred so fast after the acute optic neuritis event. We measured the next sort of a baseline time point, and then the next time point was 30 days later. And by that 30 days after onset of optic neuritis, the ganglion cells had almost suffered as much loss as they were going to. And so we tend to think of axonal loss, neuronal loss as something that happens continuously and slowly, but it may be episodic, especially in fairly acute episodes like optic neuritis. And I think timing is everything. As we learn from neuroprotective trials and these other diseases, like you mentioned, like stroke, you have to get in there really fast. And so I think that we've learned things from the upacinumab trials. I think we know that nerve cell loss occurs quickly. We know that neuroprotection and remyelination are still possible, but we need to look for new mechanisms. And it may not be a silver bullet. There may not be a single molecule that we can use. This may be a combination of different strategies that we need to use in patients with MS. Our existing therapies that attack acute inflammation, perhaps some neuroprotective or repair strategies, and perhaps therapies that are purported to work more in the innate immune system as well.
And you mentioned the animal models. The beauty of the animal models are that they're pure demyelinating, whereas in the MS brain, we have axonal loss as well. And that's one of the things that a hurdle I think we need to get over. We need to make sure we're not trying to remyelinate lost axons. And that's a confound on doing these studies, and perhaps either physiology or some of the advanced MRI metrics can better identify those pathways that have axons that need to be remyelinated. Now,
Yes.
you mentioned a bit about the innate immune system. And so tell us why you mentioned innate as we're talking about regeneration and repair and dealing with progressive disease.
Yeah, well, for sure. If we have a day of clinic with patients with MS, it's really heartening to know that we can see patients with early relapsing or remitting disease, and our existing therapies nowadays are so highly effective and so much better tolerated than the earliest therapies for MS. We've made such progress. But those other patients that we see in the course of a day, patients who've had the disease for some time and have evolved into secondary progressive MS, who have insidious progression of disability despite treatment with our best therapies, it's those patients, and you can think about it as maybe it's been termed PIRA, progression independent of relapse activity, or sometimes we'll call it insidious superimposed progression. But whatever you call it, you know it because the patient, and I think we talked about this last time, is having slow, steady accumulation of disability, even on our best therapies. And the pathological mechanisms that underlie that and the physiology that underlies that, I think, still remains largely undescribed, but we have some sense that perhaps innate immunity may be involved. Certainly microglial activation has been identified, and there's some hint from efficacy seen in trials of sphingosine 1-phosphate receptor modulators and in the anti-CD20 monoclonal antibodies that these mechanisms may have some efficacy in progressive MS. So we get a hint that the immune system may be involved, that these are not solely neurodegenerative mechanisms. And it's things like that that make us want to potentially tap into new mechanisms of action that may underlie progressive MS.
Remember to go to morningcommutepodcast.com/MS6 to claim your credit and evaluate this program. Working on mechanisms of action related to innate immunity, BTK inhibitors are now taking the stage and showing potential in the treatment of progressive MS. Let's rejoin our discussion as Dr. Bermel takes a closer look at BTK inhibitors.
One of the reasons why it's a topic of interest right now is because this class of therapies called BTK inhibitors, or Bruton's tyrosine kinase inhibitors, are therapies that are thought to act on B cell activation, on antigen presentation, on microglial activation, more mechanisms that are along the lines of what we think of as innate immunity. And these are being tested now in progressive MS in particular. And so the use of these things either alone or in combination with other therapies may have the potential to refine some of what we learned about how the immune system may be acting and which components of the immune system may be active uniquely in progressive MS. We're hopeful that this will help to unlock additional mechanisms. And I think we've had some glimmers of hope from early phase trials with larger ones starting.
So that brings us into some of these molecules. Let's talk about BTK. So what do we know about BTK in terms of mechanism of action?
Well, BTK is a tyrosine kinase. It's involved in the development of B cells. It's also useful, in affecting other hematopoietic cell lines. And so it's had its earliest use, I think, in some of the liquid oncology domains. And it's really been under intense study because it's a complex pathway, and the effects of BTK signaling potentially manyfold. But suffice to say, they involve things like antigen presentation on B cells. This is involved in the production of antibodies. It's involved in the production of pro-inflammatory cytokines, cell adhesion molecules, and involved in potentially microglial activation as well. All mechanisms that we think have something to do with progressive MS, though, the interaction between these cell types, between T cells, B cells, and myeloid cells as progressive MS pathophysiology progresses is probably very complicated, and it may not be the same in every single individual or in the same stage of MS. So as I think the scientists begin to sort out exactly which components of this mechanism of action may be most useful, the clinical trials continue to progress, and we're hopeful to learn how these agents might be useful in progressive MS, and of course also learn their safety profile. I think that's an important component as well.
Yeah, I think for me, the attraction is Bruton's tyrosine kinase inhibitors. The most appealing aspect is, I think, the effect on macrophages and maybe microglia in directing them towards a more regulatory type phenotype than an inflammatory phenotype. So my hope for them is actually be working within the central nervous system. There's mixed data on whether they get in or not. But that's how, but the macrophages get in. So those come in from the outside, the microglia are already there. But we have one study published and another that's been presented.
Yeah, that's right. I think there are, at last count, about 4 of these BTK inhibitors being investigated in MS. One of them published in the New England Journal of Medicine, I think this last summer, and another presented, and still at the phase of things where we're looking at doses and the effect of different doses with these different agents. They all have some unique individual effects, I think, and so when you're looking at doses, I think it's important to, in these early phase studies, make sure that we find a dose that balances safety and efficacy, and I think that that's the stage that we're at with this group of agents right now. The effect of the therapies that we've seen on relapses appears potentially similar to other agents, but an effect on disability progression we're hopeful will be in excess of what our current mechanisms of action will give us. And so I believe that we're just at the stage now where centers are signing on for large-scale phase 3 trials of these BTK inhibitors, mostly to enroll patients with primary and secondary progressive MS.
Yeah, that's the group that I'm most interested in this molecule for, I must say. Relapsing remitting, it'll be nice to have another agent, but that's not where our need is. As you mentioned, our biggest unmet therapeutic need right now is to better treat progressive disease. How about Mastenib? Did you hear their presentation?
Yeah, so this is another tyrosine kinase inhibitor. It has a reputation for downregulating mast cells and microglia, and again, focusing on this microglial mechanism and potential use in MS. And this particular agent did appear to slow disability progression in people with a combination population of primary progressive and non-active secondary progressive MS. And that's an important distinction because we do want to be focusing, we think, on the group of patients that truly have a need to benefit from this. So separating out the patients who have non-active secondary progressive disease, that is, patients who do not have recent relapses or new MRI lesions, I think is quite important as we select the populations that go into these trials, because we do have now therapies that work well on patients with active disease. This study that was reported at the MS Virtual Congress this fall, the joint ACTRIMS-ECTRIMS Congress, it tested Mastenib in, I think, about 300 patients with primary progressive or non-active secondary progressive MS. And, again, a dose-finding study and did appear to slow disability progression. So I think we're hopeful that some of these molecules, BTK inhibitors, Mastanib as a tyrosine kinase inhibitor, could potentially affect microglial activation, shift things more toward, as you were saying, a regulatory state, and slow the progression of primary or secondary progressive MS.
Yeah, it seemed to me they had a second study that didn't confirm their first though. Did you catch that when they were making the presentation?
Yeah, and that's where I think we really need a small dose finding study is not enough. You really need multiple trials in order to identify the dose that you're going to go with and then confirm an effect, because a lot of times the trials are designed to be extremely sensitive to pick up any possible effect that might be there. And so increases the importance on confirmatory trials. And like you said, I think the jury's still out on this one, unfortunately, of whether some of this is going to be confirmed or not.
So you made a great point a little earlier, and that the highlight that when we talk about the unmet need of progressive disease, it's definitely non-active progressive patients, because we've always had that issue of, what was the role of the anti-inflammatory effect on their activity, on the overall outcome, which may be why the results were so modest. That's what we were hopeful for in the biotin study, which unfortunately failed because that was a non-active progressive MS population. So let's look and see where all this, before I get to where it all fits in, I do want to mention one other thing on repair that we ought to talk about, because both our centers are involved in this, and that's cellular repair strategies using mesenchymal stem cells, especially neurally directed mesenchymal stem cells.
For sure. So this is a hot button issue. It's, the idea of using cellular therapies is of great appeal. There's a ton of interest among patients, especially those who are frankly most desperate, those who have tried everything that we have available to us in our therapeutic armamentarium, and they're still getting worse. Often see, the idea of a completely new domain opening up. Could cellular therapies, stem cells in particular, be something that opens up a whole new world of potential in terms of repairing the nervous system? And as you mentioned, both of our centers have been involved in experiments with mesenchymal stem cells. Jeff Cohen ran a large single study, feasibility study of mesenchymal stem cells that were autologously derived from patients' own bone marrow here at our center and administered intravenously. So many questions remain, though. I think the concept, the idea of cellular therapy is so appealing, but the science is still in its infancy, and unlocking, sort of each of the different variables that we need to unlock in order to make it work, unfortunately, I think is further down the horizon than many of our patients need. But I unfortunately do not think that the prospect of repairing the nervous system using stem cells is something that's available right now outside of trials, even though I know that, and I'm sure you've encountered patients who have gone places, tried to get, what's purported to be a repair therapy through a stem cell center or things like this. I personally try to advise all my patients to stick to the research realm for this one and enroll in trials so that we can advance the science.
I tell my patients, someone's charging you for stem cells, that's fraud.
Yeah, no, for sure. That's it's a plain way to put it, for sure. I do think that the other type of stem cells, the hematopoietic stem cell transplants for immune system tolerance, are actually progressing nicely. And I think it's important to separate out that type of stem cell. The autologous hematopoietic stem cell transplantation, or AHSCT, that is sort of a strong use of chemotherapy to almost wipe out the existing immune system after harvesting the patient's own stem cells and then transplanting those stem cells back to rescue the immune system, almost reboot it, and ideally reboot it without recurrence of the patient's MS or with a much milder phenotype. I think has real appeal, and there are very legitimate multi-center trials, including one right now that's called Beat MS that's enrolling patients and randomizing them to autologous hematopoietic stem cell transplantation versus best alternative therapy, or in other words, the monoclonal antibodies.
And we look forward to the Beat MS study. I think that the biggest difficulty with that approach is how poorly we prognosticate early on in the disease, because that would determine how much risk we're willing to recommend to the patient, knowing that early on, any of the medications may work. And so that will have to go hand in hand with that sort of approach. So let's look at that. You mentioned that. Let's look at how we're going to apply this, because taking care of MS patients has gotten very complicated, as is now, with the therapies we have, arranging how to do infusions, going over the safety and such like that. When we get into the horizon, when we're talking about either affecting adaptive immune system, neuroprotection, or even repair, we may have to think about add-on therapies, right? Because we have to turn off the inflammation. Otherwise, you're chasing your tail, right? For any sort of repair strategy. And so that was the case, for example, in the opicinumab trial. They had them all on, I believe, interferon and such. But that will be something we almost surely will have to do at the same time. And the complication there will be, well, how do we know which one to have people on as a base? You think, well, you put them on the most highly active therapy you can. So you turn off the inflammation, you're left with only the need to repair. But then you have the issues of drug-drug interactions and combined toxicities. And I think that those are going to be difficult questions to sort out.
Yeah, for sure. You mentioned, this concept of MS leading the way in terms of neurotherapeutics, and this is immediately what I think of. We're so fortunate to have disease-modifying therapies that work well in a large component of our patients. And so things are already complicated. And as you said, they're going to get more complicated. And as it gets more complicated, I think we're going to need to rely on things like more personalized medicine. And so already at our center, when someone's newly diagnosed and we're going to plan to start them on treatment, we acquire a kind of a routine panel of labs that we call treatment planning labs. It includes everything from a JC virus antibody status, hepatic function panels, a screening to make sure they don't have any chronic infections like hepatitis or tuberculosis that are latent, blood cell counts, and autoimmune panels, and things that we're looking to avoid as complications of therapy, things that we're looking to optimize to potentially augment the success of our therapies, like vitamin D level, for instance, test for diseases which are often comorbid with MS and have the potential to make it worse. So other autoimmune diseases in particular, like hypothyroidism, which is another cause of fatigue, similar to one of the symptoms that most of our patients with MS have. And so I think you're going to see, hopefully, that this expanse of personalized medicine, where we can hopefully analyze an MRI at baseline, look at baseline labs, be able to prognosticate what somebody's outlook is, will help to guide and highlight certain paths that a patient with MS may take. For that patient who walks in our office and is on one of the best things that we've got right now for acute inflammation and is still having insidious progression, maybe those are the patients where, as you said, they want to go into add-on trials. And maybe our add-on trials will get a little bit more brave. So whereas previously therapies for trials for Abudilast, for instance, or you mentioned the opicinumab trials, allowed patients to be on interferon, or in some cases glatiramer acetate, to control acute inflammation. Maybe the newer trials will be willing to be more brave and have patients on highly effective therapies. Now that involves potentially taking more risk, like you said, and it complicates the efficacy because we don't know oftentimes how these different treatments interact in terms of their mechanism of action. But from a patient's perspective, I think we want to be testing the most modern therapeutic strategies. And from a neurologist perspective, these are the real issues we deal with every day. And personally, I tell those patients, we tell them about any options we have, but also we tell them, you better check in at least once a year with us, because the field is changing. And we may have new research trials in 6 months or in 12 months that we don't have now. We may have drugs in a year that we don't have now. And personally, that's what gives me a lot of hope, is that this field is evolving and is changing. MS truly is leading the way. in terms of neurotherapeutic development.
Okay, well, there you have it. The horizon looks to be very interesting indeed. So thank you to our listeners. Thank you to Rob Bermel for this interesting conversation.
Thank you so much, Fred. And thanks to Project in Knowledge for putting this together.
And thank you for joining us today. Remember to visit morningcommutepodcast.com/MS6 to receive your credit and evaluate this program. This was the final podcast in our series on multiple sclerosis. If you have missed any of the episodes in this series or did not complete your CME CE activity, you can find them all at morningcommutepodcast.com/MS.